Welcome everyone. Today we're looking at classification and cladistics in IB biology. So let's get started. We're going to be talking about classification. It's actually quite simple. It's just a few concepts that you need to understand. First, why classify? Simple. Look at that image. What is that? It has fur, right? So we know it's a mammal. Then we already know its class. And because it's hierarchical, right? So we start off broadest at the domain and then the most specific down to the species. We can already know what its phylum, kingdom, and domain is. That would be much more difficult without classification, so it really helps identify species. Easy. It has some difficulties, right? As we said, species diverge gradually. So how do you know when a species becomes two new species? Well, this is somewhat arbitrary, and therefore it can never be determined objectively. As you can see here, it's really easy, right? A salamander is different from a frog, it's a different species. What about here, right? They look somewhat different, but does that mean they're a separate species? Well, in this case, they're not. They're the same species. Remember, within a species, there's a lot of variety as well, such as in humans. So again, think about this lack of objectivity and it's always going to be somewhat arbitrary. However, the advantages outweigh the limitations. Why? I hinted at this before, but not only is it easier to know what a species is, but then also you can make predictions about how it's going to function. For example, think about this animal, a feline, right? You probably don't know what the species is, but you know that it's a mammal. You will know that it has mammary glands, a placenta, a four-chambered heart, fur, etc. Why? Because in a taxonomic group, species will share traits since they have inherited them from a common ancestor. So it's very, very useful to make predictions about a new organism. How do we organize this in the taxonomy? Clades, right? Cladograms. A clade is a group of organisms that evolved from a common ancestor. Clades can have thousands of species or just a few. So I wanted to show this with the boxes. I think it helps. This is a clade, right? With two species. But then this is also a clade with many more species. Why? Because they're both groups of organisms that evolved from a common ancestor. This one in this case and this one in this case. All right, how do we classify into clades? We use the base sequences of DNA. We talked about that in the previous video. I'll link to that above. If that's not possible, then we use morphological traits, but remember that's somewhat outdated. If the species are extinct, however, sometimes you can use morphological traits. We can also use base sequences and the differences in base sequences to estimate when species diverged. This is called the molecular clock. You just make a correlation between differences in the basis of DNA, I'm talking nitrogenous bases, and time. This is quite inaccurate. Why? Because mutation rates vary immensely between organisms. There's three factors that affect mutation rates. These are important to remember: the size of the population, the intensity of selective pressure, and generation time. Generation time is the time from which an organism is created till the next one is created. So in viruses, this is a matter of seconds or minutes, whereas in humans, this takes decades. So mutations are going to vary immensely. In practical terms, how do we create clades? We use computer software. So you put all the species that you want to make a clade of, and the software analyzes the sequences to determine the differences. Important is that it uses the parsimony criterion. What does this mean? It's that it assumes that the changes were made with the smallest number of sequence changes possible. An example, look at this. Most of them have G, right? Guanine, but then a couple of them have adenine. The parsimony criterion assumes that it just was a mutation from guanine to adenine. However, in reality, this could have been guanine to thymine to cytosine, back to guanine, and then to adenine. This will not be taken into account by the computer software. Therefore, it does not prove how a clade evolved, because we will never know that, but it indicates the most probable pattern of divergence. It's more probable that it went just from guanine to adenine than through all of these steps. When looking at a cladogram, how do we analyze it? I mean, a cladogram is just a tree diagram with a number of branches. Interesting is that the terminal branches, these can represent either a species, right? This can be a species, or it can represent another clade. Cladograms can be so humongous, right? They can have millions of organisms if we go far back enough. So for simplicity, you don't want to draw the whole thing because you are normally only interested in a little bit of it. So you can just simplify it by, instead of here drawing the continuation of the clade, just simplifying it by making it a terminal branch. And then nodes are the points at which species diverged. So normally it represents the ancestral species, right, from which it split. And then the root will represent the common ancestor of all of these individual clades. Again, this in itself, B and C, with the node forms a clade, but then overall this is a clade. So this will be the common ancestor, the root, to all of them. This topic of morphological versus sequence analysis comes up a lot. Sequence analysis only became readily available in the 1990s. So before, everything was done based on morphological characteristics. Morphological just means looking at the appearance, the shape of the organisms, etc. So what this means is that once sequence analysis became available, a lot of them had to be reclassified because it was incorrect. A prime example of this is the figwort family. The figwort family is a type of plant family, and it had over 5,000 species. It was the 8th largest in the angiosperms. However, when they did the sequence analysis, they realized that actually a lot of them did not belong in that family. The appearance was deceiving, which is a good lesson to learn in general. When they did this, they had to move 5 groups over to other families, which left this family from being the 8th largest in the angiosperms to the 36th. So it's a prime example of reclassification. Let's do a recap question, see if you were listening. Why is the molecular clock inaccurate? And you have to decide why mutation rates are different in organisms. I'll give you a sec to pause, but I'm going to give out the answer, which is D, all of the above. Again, these 3 affect mutation rates in different organisms, which then determine how quickly they diverge and therefore evolve. So it affects the timing and the determination of this time through the molecular clock. And finally, the 3 domains of life. You might have been told at some point in school, I know I was, that life is divided into 5 kingdoms. This is not the case anymore. I mean, there are kingdoms, but the highest level is the domains, and there's only 3. It was first thought that it was just prokaryotes and eukaryotes. That would have been really easy, right? We looked at that in cell structure. But prokaryotes are so diverse that there are actually 2 humongous groups within them. And this was determined by analyzing ribosomal RNA. This is what forms ribosomes. Again, linking back to cell structure. So it's the archaea and bacteria that are prokaryotes, and then the eukarya that are the eukaryotes, including humans. All right, so that's the finish. There's nothing more to say here. Super easy concepts. Hopefully you understood all of this. If you have any questions, make sure to leave them in the comments, and I'll see you next week for A4.1.